Symposium. We have 6 presenters today. As part of their degree requirement, GES majors must conduct a faculty mentored research experience, write a thesis, and also orally present their findings today. So with that in mind, today's presenter are going to be sharing their research results with us and also taking questions from the audience. Some rules for today's presentation. Please keep your microphone muted unless when you get to the question time, you want to ask a question. Also, please hold all chat window comments until after the presentation has completed. After the completion of the presentation, we'll have a brief opportunity if you want to unmute your microphone and congratulate the speaker. After that point, we'll go into questions, which I will moderate. If you would like to ask a question, there's several ways you can do that. You can just unmute your microphone and ask your question. You can also type your question into the chat window, or you can try to raise your hand. That might be a little bit more clunky to make sure we can identify that, but those are your options. So without further ado, we'll get started. Glenn's going to get us rolling today because he's going to be discussing his work about climate and human migration. Glenn's mentor is Mark Glick from the Hawaii Natural Energy Institute. Glenn, take it away.
All right. So hello, everyone. My name is Glenn Arnade, and as a 2021 GES undergraduate, I present you with my research, which is the political and economic implications associated with climate-induced migration from Central America. And this literature analysis was conducted by myself, in addition to some guidance from my mentor, Mark Glick. So just as a brief roadmap, we're first going to take a look at some introduction to Central America, and then we'll look more into the political and economic background / history of the region, and then we'll shift and look more into the drivers of migration, and then we'll briefly go over my methods of the research and some results relating to employment, immigration, and population. And then lastly, we'll end off with a summary and some brief conclusions. So this research was conducted over 8 countries of Belize, Costa Rica, El Salvador, Guatemala, Honduras, Mexico, Nicaragua, and Panama. However, the research is more focused as the Central American region as a whole. And the main question to consider is how significant of a factor is climate change in the cause of Central American migration? And in order to answer this question, an understanding to the problem must be done in the realm of science, and then relating them to things such as the economy and politics, and this becomes a necessary task in order to synthesize solutions. So as we look more into the drought in Central America, with climate and temperatures increasing since the Industrial Revolution, the landscape of Central America has changed in various ways, whether it be through desertification, record droughts, or extreme weather events. And when we couple this with overproduction, which will be later discussed, we ultimately see land degradation. And with persisting droughts and unpredictable conditions, we see things such as reduced water tension in soils, limited nutrients, in addition to erosion, which reduces the ability for these lands to be cultivated for production. And this limited ability to cultivate reduces the livelihood potential of residents and the ability for them to sustain employment in addition to food security. Now, as we look more into the history of Central America, following World War II around 1960, we see a large amount of US intervention into various regions in order to promote democratic populism. And this was mostly to rival the growth of communism during the Cold War. And the US wanted to promote the free market by reducing trade barriers, especially in agriculture. And they also wanted the ability to sell American products in the region and reducing barriers of entry for American businesses to operate. However, they faced opposition around the 1970s against pro-communist group the Sandinistas. They resided in Nicaragua and they wanted a government-run economy rather than a free market, which caused the US to intervene with military force. And this initiated civil wars, which still kind of persist today. And then through the 1980s, we see the US tried to promote Democratic candidates in local elections. However, when they lost, the US withdrew foreign aid and caused wheat stocks to plummet. And this induced things such as famine, unequal land distribution, in addition to poverty. And on top of all of this, we have the Green Revolution occurring, which is the overproduction of crops in order to fulfill decreasing prices. And this led to things such as soil erosion and land degradation. Now, as we shift away from the background and history of Central America, it's important to keep in mind that the historical significance plays a large part in a lot of the drivers we see today. With climate variability leading to things such as unpredictable rain patterns, droughts, and flash floods, it's important to remember that these act in cohesion with the eventful history of Central America. And this makes the profitability of agriculture relatively unstable, especially with 24% of individuals in Central America relying on agriculture, and minimum yields lead to disparities such as food insecurity, limited income, and unemployment. And this motivates people to seek alternative means of income elsewhere, which incentivizes migration as a last resort. In terms of their options, farming populations could either move straight into other countries, or they can move into city centers where they face violence. And this causes the same populations to move into different countries as well. Now, a significant portion of this research was concentrated on the background due to the effect they have on current drivers, and statistical data was taken from multiple sources, whether it be from the United Nations, the World Bank, the Migration Policy Institute, USAID, in addition to some climate data from NASA. Employment, population, and immigration statistics were analyzed over a 5-year interval from 1995 till 2015, with 2019 added as the most recent data set. Agriculture and combined employment were analyzed to see how occupations are changing. Population and immigration were analyzed to see where and how people are moving. And then fiscal distribution was also analyzed to see how much the US is addressing the issue, since a majority of Central American migration ultimately ends up in the US. However, this will be later explored more in the conclusions. As we look at employment results, we have a graph comparing employment percentage over time. And as we see agricultural employment, which is labeled in blue, it is decreasing over time. Factors that lead to these things can be previous drivers that were discussed, such as drought, desertification, land degradation, which leads to reduced income. If we look at Central American total unemployment, which is labeled in red, it's important to note this is unemployment, so as it decreases, more people are getting employed. It is also decreasing with time, however, with the outlier of 2005 to 2010, and this could be due to things such as the Great Recession. And this shows that rural populations are moving out of their regions and seeking opportunities in urban centers or even in alternative countries. And as we look into immigration and population results, locations that rely heavily on agriculture, such as El Salvador and Guatemala, had the highest percentage of migrants relative to population size. Mexico is also a region that relies heavily on agriculture and had a relatively lower end migration to population ratio. However, they did produce the highest magnitude at nearly 12 million. And if we take a look at the right column, we do a summation and percentage of these statistics, we see that around 92.4% of Central American migrants moved to the United States. And as we continue, we see that immigration magnitude is increasing in Central America. However, it is important to understand that this could be a factor of population growth. So that's why I want to draw your focus to the right column where we compare total immigration to total population. And as we see, the rate has increased over time from 1995 till 2019. However, from 2015 to 2019, we see more of a leveling off, which could be due to policies from previous administrations. However, more data in the future is needed to confirm this relationship. Now, as I move on to the summary and conclusions, it's important to remember that migration would likely occur in the absence of climate change. Migration is mainly an economic and political issue in relation to things such as the Green Revolution, trade agreements such as NAFTA, and unequal land distribution. Climate just adds another layer to these various conflicts, which could lead to inflated magnitudes of migrants. It's also important to remember that Central American migration is a form of forced adaptation. It's not an act of luxury. Many people do not want to leave their homes and families as much as you do, as I imagine as many of you will not want to do that as well. And citizens mainly move just to secure an income and provide for their families back home. In fact, around 50% of the income earned by migrants are sent back to their families back home in the Central American region. And if we look more into the solutions, it's important to remember that many international policies are non-legally binding, which means they do not have a mode of enforcement. Therefore, enforcing things such as the Paris Climate Agreement, Sustainable Development Goals, or various compact agreements, and redistributing aid and capital into these regions could be a potential source of solutions. And then as we look into 2015, $3.8 trillion was spent by the US federal government. Only 1.31% of this was spent on total foreign aid for the whole globe. This isn't just Central America, this is the whole globe. And the three most relevant categories to climate-induced migration, which are international affairs, energy and the environment, and science, are the least funded categories. Therefore, increasing foreign aid to promote things such as climate and socioeconomic resilience through sustainable development, low emission infrastructure, or integrating business opportunities could be a viable solution as well. And making these developing countries into contributors of the global economy would allow governments to create their own solutions and share their findings abroad. And lastly, I want to voice that this is an ongoing issue that's ever changing. Even in the most recent weeks, our current vice president has voiced the same issues on this research in addition to the exact same drivers. That's why urgency is needed to take action within the Northern Triangle. And lastly, I would like to thank everyone who helped me along the way, such as my mentor, Mark Glick, and Michael Guidry, who has helped me through the full 3 years of my undergraduate degree, in addition to Letina, Heather, Matthew, and the SOAS program. And I would lastly like to thank my mother and father for sacrificing everything for my education. Thank you. And I will now.
Thank you, Glenn. You can unmute if you'd like to give Glenn some brief congratulations.
Awesome.
Good job,
Great
Glenn.
job, Glenn.
Yay.
Hey, nice job.
Are there any questions?
Yes, Glenn can take questions now.
Well, I'll ask you a question, Glenn. You mentioned this 1.3% of money coming from the United States. I guess that's federal money. Do you know what if there are any other significant sources of funds going to foreign countries to deal with issues in those countries?
It was actually 1.31% for the total foreign aid. And in my paper, in my actual thesis, I did a full breakdown of which departments were funding which areas in each specific region. And the areas that were being funded the most was the Department of State in relevance to immigration.
Yeah, well, I'm thinking about something other than the federal government.
Mm-hmm.
Do you know? Go ahead.
Oh no, go ahead. I didn't get to hear the ending point.
Yeah, I'm thinking of sources of funding flowing from the United States to these other countries that are not coming from the federal government.
I didn't look too much into that. I will be honest in saying that I would probably think more around into NAFTA and just business and economics and just like international business in general. I haven't really done much research into that, however.
Yeah, well, it turns out one of the big sources of money is people from these countries working in the United States and sending money back. That's just about the biggest source of money.
Thank you. I'll definitely take more into that. Look more into that.
Chris Measures.
A very nice talk. I wondered if you, during your work, whether you looked at the movement. Oh, thank you. If you'd looked at the movement of people from Oklahoma out towards the coast during the desertification in the US in the, what was it, the 30s, I think it was. Because, you know, that was people unable to continue agriculture because of the Dust Bowl. Have you looked to see whether or not land in these areas is now no longer under agriculture that it was before? Is that what's one of the driving factors?
Yes, that was one of the things I wanted to kind of look at, was something similar to the Dust Bowl. And the other thing, the other example I also thought of was Syria, with a lot of large amounts of droughts. A lot of people weren't able to produce any more crops, and that led to violence in the region. And this was kind of a look into a neighboring country that is going through, I guess, the beginning stages of a similar process.
Thank you.
Okay, we have time for one more question.
I think Rosie has a question in the chat.
Okay, oh, let's see. So have you looked at the effect of multi-annual climate phenomenon, for example, El Nino, on the region as an interacting factor?
Oh, I have not looked at I have not looked at things such as El Nino. However, that is something I would definitely look into in the future, especially as the issues start to arise more and more as climate change is up-ramping, and especially since how the magnitude and effect of El Nino could have a higher potency in the issue.
Oh, sorry, Jim Fatimra?
Yeah, thanks. Nice talk, Glenn. I'm curious if you also looked at, you know, I'm not sure if it's true in Central America, but I know in some places, as the farmers get displaced from the agricultural land, they do things like burning down rainforests and such, which exasperates the climate change. So you get this positive feedback. Did that factor in at all in your study?
Not in my research. However, I do understand that that's a thing more into cattle production as well as and doing things such as cattle grazing and more land in order to support some of those, I guess, industries.
Thanks.
Yeah, Glenn, one last point I'll make is that historically, for example, the collapse of the Maya is somewhat tied to changes in the ITCZ that's driven by changes in climate. And you can sort of see the writ large civilization doesn't quote unquote collapse at the same time. It kind of goes off in different areas at different times because of one of the thoughts is drivers of drought. So if you were to kind of build off on this, some of that work might inform, you know, what you're talking about here, at least from the climate perspective.
Definitely.
Well, great job, Glenn. Well done. We'll move on to our next speaker now. Tina's going to be sharing her research on algae on the windward side of Oahu, and her mentor is Celia Smith from the botany department.
There we go. Can I see a thumbs if you guys can see my screen? Awesome. Okay. Aloha kako, everyone. Aloha, friends, family, scientists. I'm Tina T. Huynh-Nguyen, and my mentor is Dr. Celia M. Smith. And today I'll be sharing about my GES thesis project on characterizing nitrogen and δ′⁵N values in the blade tissues of a windward population of Arnade lacerata and invasive green alga. Arnade lacerata is an invasive species to the state of Hawaii and has been the focus of statewide concern. It is a unicellular perennial alga that has proliferated all around Oahu as well as some parts of Kauai. Looking at figure 1 on the left, even though it's a unicellular organism, it's capable of displaying complex structures such as blades and a holdfast. Looking at figure 2 on the right, even with extensive manual removal, Arnade is a invasive species that is proven to be a challenge for environmental managers to manage. And in subsequent years, even with extensive removal, they have been able to reestablish themselves on a reef as observed in Moalua Bay. Other studies have documented that Arnade is able to alter benthic structures, decrease biodiversity, and is considered unpalatable to native herbivores such as our native urchins or fishes. And to make the matters even worse, it is predicted that a quarter of all coastlines in the main Hawaiian Islands could potentially be invaded by Arnade as our climate continues to warm. So what makes it so successful? The invasive succession of Arnade may be partly due to its ability to harness nutrients such as nitrogen. My objective is to characterize seasonal changes in tissue nitrogen in the blade tissues of a windward population of Arnade lacerata using 2 specific methods. First is looking at percent nitrogen, looking at the nitrogen concentration being integrated within the tissue. So if we see higher elevated percent nitrogen, we would expect or assume high nitrogen availability within the environment. Our second method is stable isotopic analysis using δ′⁵N to study the potential origins of these nitrogen sources. And to understand that, I'll be referencing values from other studies conducted here on Oahu. So a bit of background information about stable isotope is that certain ranges are associated with certain nitrogen sources. So looking at figure 4, all of these values are collected from different studies conducted here on Oahu and are not values that I've conducted myself. So looking at figure 4, on the x-axis, I have δ′⁵N. On the y-axis, I have nitrogen sources. Looking at the green bar, this is the range that we're referencing for natural nitrogen sources, referring to data collected from Ka'ena Point, and this range is from 0.5 to 2.8, which is relatively low. And moving on to the yellow bar, this is our range for synthetic inorganic fertilizer, referencing results collected from Piko Reef, and values go as low as -0.4 all the way to 1.8. And why we are able to see a negative value is due to the fact that during the production of synthetic inorganic fertilizer, atmospheric nitrogen are being incorporated within the product, therefore giving it a lower isotopic range. While looking at wastewater, we see and expect more elevated δ′⁵N values. And for our study, we're referencing values greater than or equal to 6. And that is because within wastewater, microbial processes preferably metabolizes 14N over 15N, therefore giving it a heavier isotopic value. It's also important to acknowledge that it's less likely for macroalgae to fractionate towards a specific nitrogen isotope in low nutrient environments such as here in Hawaii, and that several studies conducted here on our islands were able to successfully discern between natural and anthropogenic nitrogen sources using algal δ′⁵N values as environmental assessment tools. So finally, digging into my research, my study site is at Kolo Regional Beach Park. In the year 2020, I did a seasonal analysis looking at the wet and dry season. We predict that elevated N values will be observed in organisms located near the public septic restrooms and are denoted in the red circles on figure 5. In addition, looking at figure 5, I have a total of 12 sampling sites. Sampling sites 1 through 6 are located 25 meters from shore and I reference as my near sites. And sampling sites 7 through 12 are located 150 meters from shore and reference as my outer sites. At each of my 12 sample sites, I did plant collections in 3, and then at certain sites denoted in the blue circles, we did water column collections in 2s, looking at dissolved inorganic nutrients. After processing our water samples, we did not observe any significant difference or high concentration of dissolved inorganic nutrients within our samples. So for the remainder of my talk, I'll be discussing more about my results relating to the tissue nitrogen. My top table is data collected from the wet season. My bottom table is data collected from the dry season. So first, looking at the wet season, we see significant difference between the near site and the outer site, where it's significantly higher in the near site with values of 4 compared to the outer site with a value of 3. However, this does tend to switch when we look at %N within the same tissue, where we see a higher %N in the outer sites versus the near sites with values of 1.41 and 1.21. So looking at my dry season results, we see once again that same pattern of seeing elevated isotopic values in the near sites compared to the outer sites with values of 3.9 and 2.4. And we see that relationship being switched once again, where we see higher %N in the outer sites versus the near sites with values of 1.28 and 0.91. And these seasonal differences may be the results of increases and decreases in rainfall as well as water motion. So let's dive in deeper to my wet season. In figure 6, on the far right in the legend, these are values that represents mean delta 15N per each sample sites. And these values range from natural to mixed. On the far right of my slide, this is a small table that are data and those references that was discussed earlier in my talk. So looking more, we can see that at all near sites except sample sites 5, we have isotopic values that range between 4 to 4.6, which is most likely indicating mixed nitrogen sources. And these mixed sources may derive from the public septic restrooms, Kulo Ranch across the street, or even the cesspools, the private residentials that are operating on cesspools northwest from the beach itself. In addition, we also notice and observe potential hydrological channels, sand channels connecting sample sites 1 and 7, as well as sample sites 5 and 11. And this is more evident during the wet season due to it being potentially having more water motion and rainfall interaction during the season, which we do not see in the dry season. So when I go on the next slide, you folks will see that we don't see those elevated values at site 7 and site 11. Instead, we see a clear distinction between the outer sites, which have values that indicate natural nitrogen. And interestingly, we also see a potential variation within our near sites, sample sites, where this is evident potentially due to the concentration of nitrogen being delivered by groundwater and being more evident during the dry season, where we see less water motion as well as rainfall interaction. So in conclusion, even though we did not observe any values that indicated wastewater, that could be potentially due to lower use at Kualoa Beach Park during the COVID-19 pandemic, where park access was restricted and tourism is also at a decline, so less use of the beach overall. In addition, blade tissues collected from both seasons remained fairly low, being less than 1.5%, suggesting that during the collection period, a low volume of N is entering our reefs. Ivanvilia is more likely to reflect a combination of fine-scale nitrogen sources that ranges between natural and mixed N ranges, and that future studies on Ivanvilia should consider nutrient partitioning between the blades and the holdfast, because looking at the organism, the blades and the holdfast are interacting within different parts in the environment itself, where the blades is more exposed to the water column and the holdfast being more exposed to the groundwater. And these differences are significant, which was observed in a study conducted by Albright in 2017. I just want to thank my mentor, Dr. Celia M. Smith, for welcoming me into her lab, as well as my fellow LIMU lab mates. I love you guys so much. And I just want to thank you, Dr. Guidry, as well as Dr. Craig Nelson, and my thesis reviewer, Dr. Cynthia Hunter, for supporting me throughout this process. I also want to thank the undergraduate research opportunity program for funding my GES thesis, as well as my lovely GES program and friends who had helped me endlessly in the field and in the lab. And once again, I want to dedicate my research and talk to my mentors and those strong, powerful female scientists before me, Dr. Celia M. Smith, Mackenzie Manning, Dr. Wendy Kuntz, Colleen Allen, Nursa Brown, and Flo. I would not be here without you guys. And I'm ready to take your questions. Thank you.
Fantastic job, Tina. Please feel free to unmute and congratulate Tina if you like.
Great job, Tina.
Good job, Tina.
Good job, Tina.
Good job, Tina.
Great job, Tina.
Hey, good job.
Good job, Tina.
Good job, Tina.
Okay, we're ready for questions.
Tina, I have a question. Have you looked at either a thesis or dissertation that was done, gosh, probably 25, 30 years ago by an oceanography student who did something very similar in Kaneohe Bay. In that case, she was interested in stream inputs versus the water coming over the barrier reef. But she found very similar results. That the impact of the streams was very apparent, but it was in a very short distance from the stream mouths. And beyond that, it was all the water coming over the barrier reef was the source of the nitrogen. Have you come across that document?
I do not believe I've come across that. Was that more towards water isotopes, or was the student primarily looking at algae isotopes?
It was, well, it's obviously been years. I was on the committee. I don't remember all the details, but I just think she was doing N15, and that was one of the things that fell out of the dissertation or the thesis, that
I see.
most of the nitrogen, the source of the nitrogen for these algae was coming over the barrier reef, except, right in the immediate vicinity. And you might look that up. Marlon Atkinson was the committee chair.
Got you. That's very interesting. Thank you so much, because Kualoa is located in Kaneohe Bay, but it's on the more like end of the bay itself. So that's very interesting to look at.
Yeah, this would have been more closer to Coconut Island.
Oh, I see. Gotcha. Thank you
So,
Yeah.
for
Can I
sharing.
jump in for just a second? Excuse me, Ed, was that Allison Chun Smith? Does that name sound familiar?
Yes, it does.
And because I read that thesis too, and I don't know where my copy is, but it could be very relevant to this study. Thank you.
Yeah. Thanks, Celia. I didn't remember the student's name, but I sure remember the thesis.
Let's see, other questions. Somebody has their hand raised.
I have a question.
Go ahead.
Hi, Tina, it's Kui.
Kui.
Hi. Do you know if, if you or if Celia had anybody else look at the Delta N15 from before the pandemic as like a baseline or comparison to during the pandemic?
Unfortunately, I believe I'm been the one that has been looking at this specific site at Kualoa, just because Ivanvilia has been documented in several other areas on Oahu, but Kualoa has been a recent, discovery. So I wanted to look at the Ivanvilia population over there. But with that being said, my site is also, I think I have an image here for all those beautiful questions you guys have. My site specifically, so this is a paper done by Dan Amato and other researchers in 2019. And looking at his study, they didn't observe my site to be a site of concern for wastewater pollution due to cesspools and septic tanks. So that's why it's very important to actually look at these fine studies, because little did we know that homes adjacent to my site itself were operating heavily on cesspools and septic tanks. So it'll be very interesting for someone to continue on this research to see what it looks like on a typical year, as well as to sample these sites closer to those private homes that are operating on these cesspools that are considered a high risk, high environmental risk.
Agreed. Good job.
Tina, there's
Tina.
in the chat window, Rachel put a comment, and maybe I'll turn it into a little bit of a question. She said basically interesting to see where along that stretch of beach there might be submarine groundwater discharge, and whether the nearshore sites showing mixed values during the dry seasons might have been near the sources. Do you have any sense of that?
We were very interested to see if we will pick up, evidence of like a gradient kind of pattern with isotopic δ′⁵N. However, I believe in my study, we did not observe submarine groundwater discharge specifically. Maybe that could be potentially due to the sample season during that time. But then I also want to mention that during this time period, we did observe some variation between the near sites where there's a higher δ′⁵N between these sites. But then if I set it up differently where we extended further out into a reef, hopefully we'll be able to document and do more fine scale sampling between my two sites in itself, because my near sites was 25 meters from shore and my outer sites 150 meters from shore. But if we break it down more incrementally, maybe like 10 meters or so, or 20 meters, maybe we'll be able to observe more fine scale gradient relationship with that.
Right. Good answer. Okay. Any last questions before we move on?
I had a question.
Yes, Eleanor.
How does your data compare to the Mauna Loa Bay population?
Oh, beautiful question, because I didn't get to cover that in my talk. Thank you, Eleanor, for your question. So as I discussed earlier, I studied the Adventvelia population at Kualoa, which is on the Windward side. And a previous student actually looked at the same species, but at Piko Reef. And we did a comparison looking at their raw data. So on the x-axis, I have δ′⁵N, and on the y-axis, I have %N. And looking at that so far, and this is just our raw data, collective distribution between these two values at both of these sites best fit a polynomial relationship. And that potentially this suggests is that the alga may be able to perform at low N concentration and is adept at acquiring nutrients from various N sources. Thank you for your question. I really love this graph too.
All right. Well, thank you very much, Tina. We're going to move on to our next speaker now. Our next speaker, Mickey, she's going to be sharing her research on lipids, and her mentor is Amy Moran from the biology department. Mickey.
Okay, thank you for that introduction. My name is Mickey, and today I'll be presenting to you my thesis, which is thermal tolerance of Siphonaria normalis embryos on Oahu, Hawaii. So I'm going to start by introducing my species, and I just want to clarify that this species is not the consumable opihi that most people are probably familiar with. They're actually not related at all. So Siphonaria normalis, pictured here on the right, is a species of false limpet, also commonly called the false opihi, which means it's a type of air-breathing snail. And they reside in the high intertidal zones of Hawaii's rocky coastlines. And they're rather abundant on our coasts, both in the main Hawaiian Islands as well as throughout French frigate shoals. However, while they're abundant, this particular species of Siphonaria is rarely studied, so there's actually still a lot about them that we don't know. Looking at the global distribution of this genus, we see that species have a widespread global distribution as shown here. The species that I focus on in my study can be found in many regions throughout the Indo-Pacific, but they're also native to Hawaii, which is where my main focus will be. But why are these limpets important? So as an abundant species of herbivorous grazer, they have an important ecological role in our intertidal zone by consuming organisms. And they consume a wide variety of organisms, such as foliose macroalgae and microorganisms such as microalgae and cyanobacteria. And since they do have such a diverse diet, they have an impact on the algal assemblages by keeping species under control and preventing possible monopolization of a species in an area. So now I'm going to shift focus onto the earliest life stage of this species, which is what I studied for this project. To reproduce, the adults deposit these gelatinous masses into the upper intertidal and attach them directly onto the rocks that they live on. These masses are typically only a few millimeters to a centimeter long, and they contain dozens of embryos. So if you zoom in, here's what the embryos will look like on their first day. And over the course of about 14 to 20 days, they will directly develop within their mass, and by the end metamorphose into the encapsulated juvenile form of Siphonaria normalis, which is shown here. And at this point, they will have grown a shell, eyes, and foot before they hatch. So for my project, I wanted to examine the impact of thermal stress on Siphonaria normalis embryos. And this is because they have a pretty stressful environment where they live, where they're subject to high wave action, as well as very hot temperatures that happen directly on the rocks where they live on when the tide is low. So according to these 2017 data loggers that were placed in rocky Siphonaria habitat around Oahu, current intertidal temperatures can already fluctuate from about the low 20s, which you see over here, to about the 50 degrees Celsius. So this rocky habitat that they live in does already get very hot. And for this study, I just want to point out that temperatures were recorded twice an hour. And on the x-axis, every time point is equal to 30 minutes. So every 1,000 points takes place over about 21 days. So you can see how it fluctuates over that period of time. This data was provided to me by Shishido et al. And with climate change, this is expected to get worse because the intensity and the frequency of these hot temperatures will probably be increasing in this environment. And while the adult Siphonaria have mechanisms of protecting themselves from this heat, the embryos don't have these same capabilities. They just have to develop wherever they're laying and endure the heat before they're able to emerge with their shells and crawl around. So this is what piqued my interest in the embryonic stage and led me to my research purpose, which was to assess how thermal stress during the early development of Siphonaria normalis impacts survivorship and developmental success into the encapsulated juvenile form. This is the site that I used to collect my limpets from. It's a rock retaining wall located off of Koko Marina Center in Hawaiʻi. I collected adult limpets here, and if you look closely, you can see them. They're pretty small, so they're hard to see. I collected them between July 2020 and March 2021, and I brought them back to UH to create a limpet farm. For the limpet farm, I used a setup based on protocols and equipment from the Moran and Marco labs. Essentially, you can see the setup with this diagram. The limpets are placed in a recirculating aquarium and spread out across multiple cages, which are covered with a mesh lid to allow oxygenation but prevent their escape. And then these cages were elevated just barely above the water line on rocks and used bubblers that gently foamed water from below to keep the animals moist, because they are air breathing, but they have to be kept moist a lot of the time. And using this culture method, I checked on them daily and inspected these rocks for egg masses. After finding an egg mass, I would immediately and individually expose them to my temperature treatments by placing them in an incubator. Based on that earlier Shishido et al. Data that I showed you that recorded the real field temperatures over time, I based my temperatures and duration to be 24, 35, 40, 45, and 50 degrees Celsius for 2 hours. To monitor their development, I used a camera attachment to a dissecting microscope, and I would take these videos so that I could shift focus from top to bottom and capture as many embryos developing as possible. And this is how I was able to determine what stage the embryos were in, as well as measure their survivorship. So for each egg mass, I took an initial count of the embryos from the day 1 imaging, and I used that data as the total number of individuals within that mass. Then I would follow the progress of their development by taking these videos continuously over every 2 to 4 days. Here I have a guide showing healthy examples of different stages throughout the embryo's development. So this first image on the top left shows an uncleaved zygote on the first day after it was lain. Next, it shows the zygote undergoing cleavage, where you can see cell division happening. In photo C, we have what's called a velager, and this is where the ion shell formation begins, as well as a structure called the velum. And then this velager is a precursor to photo D, which is the encapsulated juvenile stage that I referred to earlier. And for the sake of this experiment, if an embryo had developed into this photo D, then that embryo was considered a survivor. But if development showed any abnormal growth resulting in death, then they were considered dead embryos. And that's what I defined here. I defined my criteria for these dead embryos for my experiments. These often happen in combination with each other, but I would consider them dead if any of the following occurred. So if blebbing occurred, which is where the membranes of the cell start to bulge and separate from one another physically, creating these blebs. Also, if an embryo started to discolor from these healthy greens, yellows, and browns that we've been seeing to these pale whites and blues that you can see starting to happen here. Finally, if the embryo developed abnormally, so for example, failing to develop certain structures like the shells, the eyes, or a velum, and just no longer continue to develop, I would mark them as dead. And here's a graph depicting my average percent survivorship of these embryos by temperature treatment. So for this experiment, I evaluated the survivorship of 1,545 embryos total across 21 egg masses. And you can see that after 40 degrees Celsius, there is a pretty drastic drop in the survivorship. And here I would also like to bring attention to this 45-degree treatment that contains a relatively large error bar. So this is because only one of the 6 masses contained any survivors at all, and we will discuss why that might be later on. And then finally, by the 50 degrees Celsius treatment, there were no embryos in any of the egg masses that survived into that metamorphosed juvenile stage. So while I was carrying out my normal temperature experiments, I noticed that embryos that were dying during these experiments would develop into different stages before they died. So for example, some would die during that cleavage stage, while others would make it later on into the velager stage, but they would still die. So I wanted to see whether the magnitude of temperature had a relationship with which stage these embryos were reaching before they died. So for this data, only egg masses that had dead embryos were included. I assigned a value to every embryo that had died, depending on what stage it had reached, and then I calculated the average value within each temperature treatment. And to express that visually, here's what I found. So you can see that at low temperatures up to 40 Celsius, a majority died later on at this velager stage. But then at 45 Celsius, we start to see a few more individuals dying earlier on. And then by the highest treatment, all embryos died at this cleavage stage. So they didn't make it past cell division. Overall, we can see that Siphonaria normalis could experimentally tolerate a 2-hour exposure of up to 40 degrees Celsius with near 100% survivorship. However, there was that very low survivorship at 45, and going back to that single egg mass of 6 that had survivors, this may suggest that the temperature that some masses can tolerate are higher than others, possibly due to a genetic component from previous generations that help increase its thermal tolerance. And again, by 50 degrees Celsius, there were absolutely no survivors. And these upper thermal treatments are very similar to temperatures that are already common in the field habitat of these animals. So from this, we can see that at least in some areas, current Hawaiian intertidal temperatures are already high and long lasting enough to impact the survivorship of this species. And this is something that most likely will be worsened by climate change as average temperatures continue to increase. For the stage reached before death under different temperature treatments, this area of study may provide insight on the specific developmental mechanisms that are being disrupted by thermal stress during early development. So for example, at high temperatures, development halted at the cleavage stage 100% of the time, which may suggest that these high temperatures are messing up cell division and no longer allowing them to continue. But then at these mid temperatures, most embryos were still able to continue into that later velager stage before they died, but they still ended up dying. And that suggests that these temperatures are still lethal, but for a different mechanism that we're not sure of. But overall, thermal tolerance studies of ecologically important species is necessary to anticipate future risk to these species under warming conditions, as well as predict the resulting potential changes in our Hawaiian intertidal ecosystem. And with that, I would just like to acknowledge some people for assisting me with this project. Dr. Amy Moran, my mentor, for providing me advice and support, and all of my friends and family, the GES department, and my good friend Devin Kochi for assisting me in my Olympic collection. And thank you all for attending this presentation as well. And with that, I'll be happy to answer any questions.
Thank you, Don, Mickey. Please feel free to unmute to congratulate Mickey.
Great job, Mickey.
Good job, Mickey.
Yay.
Thank you.
Good job.
That was very good, Mickey.
Nice job.
Thank you.
We have some time for questions.
Mickey, I have a question. I'm intrigued that the temperatures are getting that hot, that 45 degrees, holy cow.
Yes.
That's obviously not the air temperature, so it must be the water temperature. So how deep is this? Water must be awfully shallow, yeah?
So it's actually the temperature that's happening directly on the rocks, which is why it's getting so hot, because these limpets, they live in the upper intertidal. So they tend to be on the higher rocks of our coasts. So yeah, it's directly from the rock temperature that it's getting that hot.
Okay, now 2 hours, that's acute toxicity as the EPA would think about it versus chronic. How long does it normally take these organisms to move from embryos to whatever villagers and so forth?
So according to my data, it would take about anywhere between 14 to 20 days for them to actually emerge from their egg mass. And that's the point where they actually have like their shell cap and they can crawl around. But prior to that, they're still developing in that gelatinous mass that I showed before. Yeah.
Oh, so it's conceivable that even a lower temperature, if they were exposed to it for 14 days, would do serious harm to them.
Oh, absolutely. That's something that I think future studies would really benefit with, because my study, I only exposed them one time to these temperatures within the first 1 to 2 days of their development. So if you even consider like a mid-range temperature or a lower temperature multiple times, I mean, that could go either way, where it could increase their tolerance or it could, weaken them with repeated exposure. So we really don't know how that repeated impact might have.
Okay, thank you.
Chris, measures?
Yeah, that was a great talk. I was curious about the 2 hours because, the tidal cycle is such that many of these would be exposed to the air. In other words, the rock would be heating up as the tide goes down before the tide comes back in again. Have you had a chance, or might you look into sort of the integrated time of exposure? Because I suspect it might be a lot longer than 2 hours, given the tidal cycle.
Yeah, so I wasn't able to incorporate the entire fluctuation of temperatures into my project. But yeah, so according to these loggers, these high temperatures would be maintained for about 2 hours before they would drop back down again. And so that's where I got my duration from. But it definitely is much more complicated in terms of these fluctuating temperatures when the tide is low.
Hey, Mickey, I have a quick question. Did you notice any changes in the behavior of the specimens at the different temperatures?
That's a good question. Not definitively, I would say. I mean, I could tell when an egg mass was not doing so well at some of these temperatures. You can see that they're still developing into the velager, but like I said, they have these blebs happening and they're kind of looking wonky, and that was mainly the most visible thing that I've seen.
Kui has a question. Kui?
Hi, Nikki. Good job.
Thank you.
Okay, have you looked at seasonal differences? Like, did those loggers log throughout the whole year? and if they did, were there seasonal differences?
They did log throughout the whole year. So the snippet that I showed you started in August, so when it would be pretty hot. I did not look too much over the entire year. I kind of just wanted to pick where like I saw these maximums throughout the year and simulate that in my experiments.
Okay, how often do these guys spawn? Like, do you know if the hot season would be when they're larvae or if it's the cool season?
It seems like, well, at least with my experiment, it seems like they spawn year round because I've been collecting limpets for a full year and I would get masses throughout the entire year.
Okay.
And I believe they spawn once or twice every year per adult. So I assume that any temperatures experienced throughout could potentially affect these embryos.
Okay. Awesome job.
Okay. Any other questions for Mickey? All right, great job, Mickey. Well done.
Thank you.
All right, our next speaker, Eric, he's going to be sharing his research on deep sea floor scavengers. Eric's mentor is Dr. Jeff Drazen from the Oceanography department. Go ahead, Eric.
Sorry, I'm getting it set up. Oh, hang on. Okay. Oh, it's on the wrong side. Okay, so hey everyone, my name is Eric Klingberg, and for my thesis, I'm looking at linking ecological conditions to size distributions of abyssal scavengers, and my thesis mentor was Dr. Jeffrey Drazen. So the abyssal realm is the most expansive habitat in the world, stretching over a majority of the ocean, as you can see from the gradient areas of this map, at depths around 3,000 to 6,000 meters. It's a place with no sunlight and immense pressures, yet it supports a diverse community of marine life. And they feed on the particles and the dead organisms that sink from the upper ocean. However, due to the logistical challenges and the resources required to study these remote locations, very little of the abyssal realm has been explored. And so scattered across the abyssal plains, there are these small metal concretions known as polymetallic nodules that are found along the seafloor, as you can see from the background. And these nodules contain metals that are found in consumer electronics, as well as the batteries that could be used for clean energy storage in the future. So there are plans underway to mine these regions for those valuable resources. However, the impacts of these operations involve kicking up the sediment and wiping out the organisms that live on the seafloor, decreasing the biodiversity for decades and beyond. And additionally, the discharge plumes from these operations would negatively impact midwater communities and could introduce toxins such as mercury into the food web, contaminating the seafood that we eat. And so the Clarion-Clipperton Zone is the region south of Hawaii towards off the coast of Central America. And it is estimated to have around 30 billion tons of nodules on the seafloor, as you can see from the reddened areas of this figure. And because of this, there are already mining claims in place, as you can see from this figure with the mining claims. And there are also areas of particular environmental interest, or APEIs, where no mining would take place. And so because of the plans to mine the CCZ, there's a body of research studying this ecosystem prior to the start of mining. And this is essential for measuring the environmental impacts that these operations could have. And so for my thesis, I looked at the scavenger fish community that live in these waters. And so these are the fish that are known as rat tails or grenadiers, but specifically 𝘭𝘢𝘕𝘓𝑚𝑑𝑒s and 𝑢HM. And they're highly prevalent at abyssal depths and occupy the top trophic level as opportunistic feeders. And these rat tails feed on the benthic organisms on the seafloor, as well as the carrion from large fish that sink to these depths. And by assimilating the carbon and nutrients stored in those carcasses, they effectively sequester carbon into their bodies. And as they forage across the ocean, their messy eating style, as you can see from this video, in addition to their defecation, provides nutrients to lower trophic levels, so they serve an important ecological function. And so the goals of this research were first to compare the size frequency distributions of 𝘭𝘢𝘕𝑚𝑑𝑒 species from the CCZ to Hawaii. And next to identify what conditions could explain those observed size trends by looking at different environmental parameters. And lastly, to look at the seasonal changes to Hawaii, where we had different study sites that took place during different times of the year. But first, why is it important to study fish size? So fish size is indicative of the life history of a fish, from its age and reproductive potential to where it fits into the food web, because their diet shifts to different sources as the fish grows. And fish size is also sensitive to anthropogenic impacts such as overfishing and pollution. So baseline ecological studies are needed to measure how activities such as deep sea mining could affect these ecosystems by looking at fish size. So the methods for studying scavenger length involve the deep cam system. And this is a baited camera approach that allows for observations of bait attending communities and their natural environment. And this is done by creating an artificial food fall with a deployment of the system that has bait attached to it, as you can see from the figure. However, one drawback to this is that you cannot identify the species to a precise level because they aren't being sampled. And so in this case, it was not possible to distinguish between 𝘭𝘢𝘕𝑚𝑑𝑒s and 𝘈𝘕𝑚, which are two different species of rat tails. And so the deployment recording sequence is as shown. And the resulting data was placed into the program event measure. And the video stills captured by the video footage were put into the system. And the preanal fin length, or the PFL, was the length that was used to measure these fish. And it's the length from the snout to the pectoral fin insert. And this was done because when these fish are collected, they're often missing the tail tip, and their movements make it very difficult to measure the entire length on camera. And then for the statistical analyses, non-parametric comparisons were made to look for significant differences in size between the study sites. And then a generalized linear model, or GLM, was constructed to try to explain those differences in sight with different environmental parameters. And so this is the deep cam deployment sites. And they took place over different cruises over different various years here in the CCZ, and then at Station Aloha, which is a site that is north of the island of Oahu. And so the size distributions are shown here with this violin plot, with the preanal fin length on the y-axis and the location or the stratum on the x-axis where those deployments occurred. And the thin dotted line here is the mean size, and then the thin solid line here is the median size, and then the boxes are from the lower to the upper quartile. And so, but for the results, it was shown that in the CCZ, the fish in the eastern CCZ were significantly larger than those in the west. And then the fish that were observed at the two abyssal hill deployments, at ECPAC 2 and UK1 hill, were larger than their respective planes, which was first observed by Leitner et al. 2017. And then surprisingly, at Station Aloha, the fish that were seen during the winter at this CAM 2002 were roughly twice the size as the fish observed during the summer. And in fact, the winter fish were statistically indistinguishable from those seen in the Eastern CCZ, which I will talk more about later. And to help visualize this, here are the deployment sites again, with the fish length and what I like to call the fish scale, the larger fish in the east and the smaller ones in the west. And so to explain the differences in size, the different environmental parameters were plugged into the generalized linear model. And some of these have been shown to influence rat tails in prior research, such as Leitner et al. 2017. And so these included proximity to local bathymetric highs as measured by the BPI. And this was because proximity to local highs has been shown to increase the transport of food particles to organisms that rat tails can feed on. And then for the percent nodule cover, nodules can serve as a substrate and a hiding place for benthic organisms, which may increase the food available to rat tails. And then the bottom temperature has been shown in that Leitner paper to influence rat tail size, and it could possibly have something to do with metabolic rates. And then the monthly surface chlorophyll A, this provides an indicator for foodability that could be coming from surface processes. And particulate organic carbon, or POC at depth, this could serve as another indicator for food for benthic fauna. And then the C4 slope, because as there's an increase in slope, it could be an increase in the POC flux to benthic communities that rat tails could feed on. However, these are the two that I want you to remember. So for the environmental parameter findings, the final GLM results showed that the mean preanal fin length was significant in terms of the proximity to bathymetric highs and the surface chlorophyll values, as you can see from the figure with the mean length on the y-axis and those important parameters on the x-axis. And so this essentially means that regions with the increased food supply featured a higher proportion of larger fish. However, interestingly, these results differed than the Leitner et al. Paper, which studied some of these regions as well. And they found that temperature and nodule cover were significantly related to rat tail size. However, surface chlorophyll was not significant. So my project involves comparisons across a broader region. And so this could be revealing the importance of large scale differences in productivity, while perhaps missing the importance of more localized conditions, such as nodule cover. And so one limitation of this research is that the deployments across the CCZ took place at different times of the year. And so it's important to consider how fish size can change seasonally by looking at the Station Aloha deployments. And fish length in the winter was twice that of the summer. And so why is this? Well, these species are known to be very slow growing. So we know that they did not double in size between those few months. So it looks likely that a possible seasonal migration is occurring. But the question is, what is happening in Hawaiian waters during the winter that would attract these fish? And so during this time, the three most common tuna species in Hawaii, the big eyes, yellowfin, skipjack, they migrate to Hawaiian waters. And so this is also when the longline fisheries are their most active. And this could result this results in tons of bycatch entering Hawaiian waters. And this would create a seasonal surge in the food that is available for rat tails and potentially explain the larger individuals that were there, because the larger fish depend more on carrion than the smaller rat tails. And so for the conclusions, there are significantly larger fish in the Eastern CCZ than the Western CCZ. And this is attributed to the increased productivity and the proximity to topographic highs that support the larger rat tails that were shown in the Eastern CCZ. And then a seasonal migration of rat tails to Hawaii could be connected to tuna migrations and fisheries bycatch. However, we only had one deployment in the summer and one deployment in the winter. So more beta camera studies are needed to examine or verify a relationship between pelagic migratory paths and the abyssal scavengers. And additionally, more studies are also needed to understand the variance in fish size. And this is especially important prior to deep sea mining. Otherwise, it will not be possible to distinguish between anthropogenic impacts and changes due to natural processes with regards to how fish size changes. Special thanks to my family, Dr. Michael Yitri in the Oceanography office, Dr. Jeffrey Jason, Astrid Leitner, Jesse Vandergrant for their help with my thesis, the data collection and the modeling, and everyone from the Deep Sea Fish Ecology Lab, Cheryl Gaspi in the COCUA program, the UOB, the GES Ohana, and anyone who has given me a ride, fed me, or helped me move. Thank you.
Nice talk, Eric. Well done.
Thank you.
Feel free to congratulate Eric before we move on to questions.
Good job, Eric.
Good job,
Eric.
Nice job, Eric.
Yeah, Eric.
Good job, Eric. I'm proud of you, brother.
All right, our first question we'll give to Craig Smith.
Hi, Eric. Excellent job. Really enjoyed it, and I enjoyed reading your thesis. I have a question. Seems to me that chlorophyll A is obviously tracking surface primary production, and POC flux is tracking food availability at the base of the food web at the sea floor, so food for benthic invertebrates that the rat tails are feeding on.
Mhm.
So do you think that chlorophyll A is a better index of large carrion that's sinking down and getting efficiently to the seafloor? Do you think that's why it's strongly correlated on a regional basis with size?
Yeah, because such a broad region was studied for my paper, what the chlorophyll A kind of indicated was where there was generally a higher degree of productivity in the Eastern CCZ than the Western CCZ, because the Western CCZ is a little more oligotrophic. And so I think it's just kind of what my results were showing was linked in relation to the general productivity of the area. I think POC actually might be actually it could be better in some cases, but I used the data for POC that I used came from a paper at Lutz et al. I think it's 2007. And that was an annual estimate of POC, but I think it would be better to use something like sediment traps. I think that would be a better measurement for productivity that's actually reaching the seafloor.
Okay, actually, but my point is that the POC flux at the seafloor is feeding the base of the food web, the polychaetes and things that the smaller seems to be preferentially ingested by the smaller fish, whereas the chlorophyll A at the surface ocean is tracking the primary productivity that's going into tuna and things, sinking rapidly down and not being degraded on the way down. So those actually may be tracking different kinds of food for rat tails, and where you have a higher surface productivity, you may have a higher biomass of large potential carrion that can sink rapidly to the seafloor and feed larger fish.
Yeah, that's a really good point.
Thanks. Good job again.
Yeah, and thank you for reviewing my paper.
Oh, no problem.
Eric, I have a question. As I understood it, you were suggesting that the bycatch from the long lining fishing was attracting visitors. That to me is provocative, that that the longline fishing industry is having that much of an impact. Holy cow. Do we have any estimate of the catch versus the bycatch associated with this operation?
Yeah, there was a recent paper that found that the bycatch actually exceeds the targeted catch for longline fisheries, which is, yeah, it's pretty.
I mean, are they trying to do anything to reduce because it's just throwing these things back into the water, right?
Yeah, for the most part.
Are they doing anything to try to change that situation? It seems like
So Ed, this is Jeff Drazen. The long-nosed lancet fish is now the highest catch by abundance, and they've tried to get the KCC culinary school to try to cook it and figure out what to do with it, and they consider it unpalatable.
Well, there's a nasty species here called nutria, which is a nuisance, and they're turning it into dog food. The dogs will eat it, so maybe that's a possibility.
Interesting idea.
Other question? Another question?
Yeah, Eric.
Yeah, Chris.
It struck me, what do we know about migratory paths of benthic fish? Do they migrate?
Yeah, so these fish have been shown to migrate across potentially ocean basins.
Oh.
And so they're kind of, and because these food falls are relatively rare events, you know, within the whole expanse of the ocean, they have to swim far distances to find meals.
So basically what we could be seeing here in your data sets is basically some sort of fish memory that they know there are certain places, certain times, kind of like the whales, of course, are migrating, you know, for the primary production hits in seasonal ones. So there's a sort of similar thing going on with the benthic organisms.
Mm-hmm. Yeah, and so I'd be really curious to see further research explore that and trying to see if they're following the migratory paths of other fish that are, you know, living at the surface, such as tuna.
I mean, that's obviously a problem with your analysis with the data, is that you're looking at parameters, but, you know, if these guys are moving around, then that's another variable that is very difficult to control for, of course.
Mm-hmm. Definitely.
Oh, interesting.
Okay. Any other questions for Eric? All right, great job, Eric. Well done.
All right, thank you. Thank you, everyone.
So our next speaker, Mariam, she's going to be sharing her research on plankton in Mānoa Bay, and her mentor is Margaret McManus from the Oceanography Department.
All right, thanks, Dr. Guidry. There's some construction going on today, so hopefully that doesn't get too distracting. But besides that, we will get started. So thank you everyone for coming to my presentation today. I will be presenting my GES thesis on the environmental influences on the vertical picoplankton distribution in Mānoa Bay of Oahu, Hawaii. So here's a quick overview of what I'm going to be covering in this presentation. I split my results into 4 sections that will cover the environmental properties of Mānoa Bay, the cell abundances and their relationships to those environmental properties, a comparison with station aloha data, and some seasonal variability, and then ending with discussion and conclusion. So the 2 picoplankton I looked at in my study were Prochlorococcus and Synechococcus. These are classified as photosynthesizing phytoplankton less than 1 micrometer in diameter and are located in the photic zone, which is about the first 200 meters of the water column. Prochlorococcus is going to be our more abundant of the 2, not just in the North Pacific subtropical gyre, but globally as well. And they tend to do better. They tend to be more abundant in oligotrophic waters. They are also smaller in size, ranging between 0.4 and 0.6 micrometers. Synechococcus is going to be our larger of the 2, reaching 0.9 micrometers, and tends to be more abundant in eutrophic waters, but will still exist at some magnitude in nutrient depleted waters. So my objectives of my research, I really wanted to focus on analyzing the influences of environmental factors on the vertical distributions of Prochlorococcus and Synechococcus, first in a nearshore setting, then in an offshore setting, still within Mānoa Bay, comparing the 2, and then finally comparing all of that to Station Aloha located in the pelagic, as you can see on the map here. So for our study sites, we had 2 nearshore sites, no further than 2 kilometers offshore at about 150 meters and 180 meters deep. And our offshore site was 7 kilometers at about 550 meters deep. And all of our samples were collected from Niskin bottles on a quarterly basis between 2013 and 2020, about a year of which I helped collect those samples with the McManus lab and in collaboration with the Carl lab. And as for our lab procedures, our picoplankton data was acquired with a flow cytometer through the McManus lab and the Seymour staff. I ran my own chlorophyll analyses, and nutrients were analyzed in the S lab here at UH Manoa, and I made all my visualizations using MATLAB. So the environmental properties I'll be covering are chlorophyll, nitrate, and photosynthetically active radiation, or PAR. So here's our chlorophyll profile for all of our Mamala base stations and Station Aloha between 2013 and 2020. Chlorophyll is going to be in units of micrograms per liter and plotted against decreasing depth in meters. And we can see how chlorophyll reaches these maximums at different depths depending on distance from shore. So our nearshore stations, about 2 kilometers offshore, had a chlorophyll maximum at about 75 meters. Our offshore station, 7 kilometers offshore, that had a maximum at about 100 meters, and Station Aloha, 100 kilometers offshore, had their deep chlorophyll maximum at 125 meters. It also goes to note that there is significantly more chlorophyll at all of our Mamala base stations when compared to Station Aloha. And this is an indication of what is called the island mass effect, something I'll get into a little later. Now for our nitrate profile, there is this consistent increasing trend with depth across all of our stations and our Station Aloha data. So we have this pink dashed line indicates the nutricline where nitrate concentrations in micromoles per kilogram exceed 0.1. And in our Mamala Bay stations, we saw that nutricline it was actually between 65 and 80 meters, so somewhere in here. And then for Station Aloha, it's going to be lower, just given to its deeper depth at a farther distance. Now for our PAR was actually collected using a PAR sensor. And from that, we saw how it followed a pretty predictable profile. We saw light started decreasing from the surface. And this graphic right here actually shows the depths at which PAR reached 1% and 0.1% of those surface light levels. And for our 1%, we saw it seems relatively the same through stations 1 and 3. When we get to station 3, that depth becomes a little lower. And then for 0.1% of light levels, that decreases in depth the further we get away from the island. And not to go into our cell abundances and their relationships to these environmental properties I just discussed. So these are box plots with the median cell abundances for both Prochlorococcus and Synechococcus with their 25th and 75th percentiles, and the plus signs indicate outliers in the data. So our Prochlorococcus distribution on the left here these are going to be in units of cells per milliliter and in a magnitude of 10 to the 5th. So in this distribution, we have subsurface maximums. In Mamala Bay, we see that maximum at 45 meters before abundances begin to decrease. And at Station Aloha, it's going to be a little lower at about 75 meters. We also saw how Prochlorococcus actually increased in abundances the further away we got from shore, whereas with Synechococcus abundances, we saw the opposite. The higher abundances of this picoplankton were actually closer to Oahu and decreased by an order of magnitude when we got to Station Aloha. Now our nearshore distribution for Seneca-Coccus just followed just decreasing all the way, starting from the surface, and our Station 3 and Station Aloha had slight subsurface maximums at about 45 meters and then 75 for Station Aloha. It's a little hard to see just because of the lower concentrations of Seneca-Coccus. Now, here are my comparison graphs with the environmental properties to cell abundances. I apologize for the noise if it's being distracting, but so our x-axis is going to be our concentrations of our parameters, and y-axis is going to be our cell abundances. And these light colors, these yellows, are going to indicate shallow depths, so up to about 50 meters in the water column, and then our darker colors are going to be deeper depths, up to 150 meters. And then from these plots here, we don't really see any relationships. We can see and kind of make out where these chlorophyll maximums happen that correspond with 75 meters, but we don't see the chlorophyll maximums corresponding or matching with the cell abundance maximums. And the same thing with Seneca-Coccus, we can kind of make out where those chlorophyll maximums are. We can indicate where the cell abundance maximums are, but the two just don't match. Now for my nitrate comparisons, I put these on a logarithmic scale to show this negative relationship between the two. So we can see that the lighter colors will be in areas or at depths with higher abundances and correspond with low nitrate concentrations, nitrate still being in micromoles per kilogram. And as we go deeper in depth and as cells begin to decrease, nitrate will increase as a result. And here at Station Aloha, we have a similar trend. However, we do have this kind of stationary area where cell counts are not changing with depth. This could just be due to some mixed layer action happening out there. And with Seneca-Coccus, we still see that negative relationship on the log-log scale. However, as we get to Station 3, it starts to become less distinct. As I mentioned earlier, cell abundances started to decrease even at our Station 3 site, and even more so at Station Aloha. That negative relationship starts to become almost unclear just because of just less Seneca-Coccus abundances. Now for our par comparisons, it's a pretty straightforward. As I mentioned, light decreases with depth. So where light levels are going to be the highest, our cell abundances are also going to be the highest. And where light levels are the lowest, we are going to have our lowest cell abundances as well. And this was seen at all three stations. All right, now to touch on some seasonal variability. So I took the overall cell abundances from Station 2 and, sorry, can you still hear me okay?
Yes, Mariam.
Okay, cool. All right, so here, so yeah, I took the overall cell abundance distribution from Station 2 and divided it into 4 seasons. And this way we can see how the subsurface maximums and surface abundances fluctuate throughout the year. And they are plotted again, or not against, but we have the mixed layer depth in the blue and the deep chlorophyll max in green and the nutricline in red, just to show these environmental parameters to see if there's any, kind of to show how they respond, if there's a response at all. So we can see for Prochlorococcus, we do see some correlation with the mixed layer in the spring and the summer. We also see our highest Prochlorococcus abundances in the summer. And with Synechococcus, we have our highest abundance in the winter, and we have a slight subsurface maximum, but not quite reaching the surface abundance here during the summer at 45 meters. And we saw similar patterns like this at our stations 1 and 3. All right, now just to wrap things up in summary, we saw how our nutrients had the most direct negative concurrence with the vertical picoplankton distribution at all of our sites, and with Synechococcus, especially at our nearshore sites, and PAR had the most direct positive concurrence just across the board. And that heightened Synechococcus presence I brought up, and the increased chlorophyll presence in Māmola Bay that's due to this island mass effect is really stimulated from land introduced runoff. So in this figure, the human derived runoff we see here, and thus it increases nutrients at the surface and increases phytoplankton productivity. And then along with mixing and upwelling, we create this more nutrient rich environment for Synechococcus to kind of produce more than you would see at Station Aloha. So these arrows are just saying we saw more Synechococcus closer to the island, and then Prochlorococcus was actually increasing the further away we got from shore. And on top of these parameters, we saw that these varied seasonally as well. All right, so to wrap things up in conclusion, our observations revealed that the island of Oahu had contributed to a variability in the pelagic ecosystem, including carbon fixation and nutrient cycling. This was especially highlighted in the environmental properties of Māmola Bay that had enhanced peaks in abundances due to this island mass effect. And we observed both similarities and differences between nearshore and offshore sites, even including Station Aloha. So that concludes my presentation. I want to give a special thanks to my mentors and my advisors, Dr. Margaret McManus and Christina Comfort, for helping me throughout my thesis, my academic work in the lab. They've been super helpful. And my academic advisor, Dr. Drazen, for even introducing me into Margaret's lab. My family and my friends for their never-ending support. And the SOAS faculty and staff for helping me along the way. I also want to give a special thanks to our vessel support, Michael for helping us out on our cruises, and the SV Ho'oponopono, the RV KOK, and the RV Kilamoana for supplying us with Station Aloha data and some help with even our own data as well. And of course, for our funding, especially SWAC A prizes for funding this thesis. And here are my references, and I'm happy to take questions.
Good job, Mariam. Feel free to unmute if you'd like to congratulate Mariam on her presentation.
Great job.
Great job, Mariam.
Awesome.
Good job, Mariam.
Great job, Mariam.
Awesome.
Sorry again for the trash truck.
Nope. No, you did. I mean, especially with that background noise, you did really well. It didn't come across too badly. So
okay,
in terms of the background noise, your talk was excellent. So
okay,
could hear you, no problem. All right, questions from the audience.
Okay, Miriam, I have a question. There's a sewer outfall in Māmola Bay, and it looked to me as if stations 1 and 2 were right in the vicinity of the sewer outfall from Sand Island. Is that true or not?
I didn't look too much into that. So the original placement of these stations was actually through the SWAC project that they want to do in downtown Honolulu, but got pulled sometime this year, I believe. And so I didn't really consider that very much, but you could argue that it does contribute from that land-based nutrients. It contributes to the runoff that is increasing this the phytoplankton abundances there.
Well, this is has financial implications because the city has been under the gun from the EPA to go to secondary treatment. And the argument is that the sewer outfall is having some kind of adverse effects. But if I were you, I would sure look at the location of that sewer outfall. I think it's right there in the vicinity of Station 2. I mean, it's right off the main channel going into Honolulu Harbor. And it used to bubble up right to the surface before you were born. But it was in shallow water and you could see it flying over, you know, landing at Honolulu Airport. That was very apparent. The other question I have is you saw this negative correlation between nitrate and chlorophyll, I guess. How would you account for that?
So the negative correlations with the nitrate, it was between cell abundances actually.
Okay.
Yeah, so I'm sorry, what was the second half of your question?
Well, how would you account, you know, why is there a negative correlation?
So just from the, so I would say it's mostly from Synechococcus needing more nutrients since there's more at the surface in a nearshore, in that nearshore environment. And Synechococcus has a shallower vertical distribution than Prochlorococcus. So when that starts to decrease, when that starts to disappear, we'll get more and more nitrate.
Yeah, well, I would say the reason there's a negative correlation is that phytoplankton are taking up the nitrate and turning it into cells. So when the nitrate goes down, the phytoplankton go up.
Right.
Okay.
The other questions?
Chris Measures has one.
Chris, yeah, go ahead, Chris.
It's sort of more of a comment. It struck me watching your data. It's interesting. Normally we think of stratification in the surface ocean as a barrier to nutrients coming from below to feed the surface water where the light is. But actually next to the island, it's exactly the opposite. The stratification brings the nutrients from the island and traps them in the surface layer because of the stratification that comes in with submarine groundwater, for example. So it's just a curious sort of contrast between those two systems. It's more of a comment than anything else. You don't have to answer it.
Yeah, no, definitely. And also the bathymetry of the area, there's like some underwater canyons there. There's very steep slopes in that sampling site. So that could also contribute to this this upwelling and mixing of nutrients to the surface. So.
Well, we did a study years ago looking at N15 and a transect from, let's say, Diamond Head to Barber's Point. And there's a very systematic pattern there that reflects the input of nitrogen coming from the land.
Right. Yeah, and especially from Sand Island, I can imagine.
Yeah.
Okay, any other questions or comments? Okay, great job, Mariam.
Thank you.
Okay, so we're at the end of our symposium. We have our last speaker. Kayla is going to be sharing her research on climate clouds and islands, and her mentor is Giuseppe Torri from the Atmospheric Sciences Department.
Hi, everybody. My name is Kayla, and for my spring of 2021 Global Environmental Science thesis project, I worked on a project with my thesis mentor, Dr. Giuseppe Torri, titled Simulating the Effects of Climate Change on Cloud Formations Around Mountainous Islands. So just to walk you guys through it a little bit, I'll start with the introduction and why this study is important, go into how we conducted the study and the methods, some of what we found and what it means and the results and discussion, and then I'll wrap up with some concluding points about those results. Looking at why this study is important in particular, islands can act as key developers of convection in the tropics. And when I say that, I mean that they can influence the atmosphere above and around them because of their diurnal cycle of convection. And when I say diurnal cycle of convection, I'm talking about a process where, in our case, we have an island in the middle of the ocean, or just a piece of land. And when the sun comes up, it begins to heat up the island, causing the air above it to rise, forming clouds of precipitation above the middle of the island. And so this typically peaks in the early evening or the late afternoon, whenever the island is at its warmest. And then the air is pushed outward, sometimes taking some of the tops of the clouds with it. It falls again over the ocean. And then at the nighttime, essentially the opposite takes place. And the land begins to cool down, and essentially the ocean becomes warmer than the piece of land in the middle. And so we have air rising above the ocean and clouds of precipitation forming above the sea, which typically happens in the early morning or the very late nighttime whenever the island is at its coolest. Then the air is pushed inward and it falls again over the middle of the island. So in this presentation, when you hear me talking about the island diurnal cycle of convection or just the diurnal cycle, this is the process that I'll be referring to. Looking at the diurnal cycle a little more, the driving mechanisms are a little unclear and lots of climate models do not represent the timing very well right now. And so our understanding is limited since there are so many factors that might influence this process. Furthermore, islands have a very large reliance on fresh water, and some islands rely solely on their local rainfall as their only source of fresh water. So understanding the diurnal cycle in a little more detail can help us understand issues related to habitability and water security in the maritime continent and in the tropics. So for our study, we explored the diurnal cycle in a little more detail using a numerical model, which allows us to study the process above and around an idealized island or sort of perfect island in model speak in great detail to give us an insight as to what might happen in a future of anthropogenic climate change and to sort of fill in the gaps of what we already know about diurnal cycles in the maritime continent and in the tropics. So we conducted this study using a cloud resolving model, also known as a CRM. Here on the right is an example of a cloud resolving model. Cloud resolving models are one of the main atmospheric simulations used for studies like these. And so they allow enough grid spacing for the simulation to develop clouds either over certain parts of the island or the whole island, depending on what conditions you set for it. So in our study, we set some very simple conditions so that we could understand the fundamental dynamics behind the process that we were looking at. We used Cloud Model 1, also known as CM1 for our simulation. And like I said, we set some very basic initial conditions so that we could really focus on the fundamentals of the diurnal cycle itself. And so we did this by creating two islands. We created a flat island and a mountainous island. We actually created a third island as well, but I won't be talking about that one in this presentation. So for a flat and mountainous island, we imposed a diurnal cycle in the initial conditions. And we have a domain of 64 kilometers with an island of 16 kilometers centered right in the middle. And then our mountain is 1 kilometer tall. So our driving question behind this study was, how does climate change affect the rainfall and cloud formations above and around these islands? And there are many ways we could have approached this question, but we decided to approach it by changing the sea surface temperature to simulate anthropogenic climate change. So we changed our sea surface temperature from 300 degrees Kelvin to 303 degrees Kelvin in 0.5 degree Kelvin increments. So that gave us seven sea surface temperatures that we were looking at. And so we took readings in the middle of our islands, on the coast, and over the ocean. So we could really compare what was going on in all areas of the domain of these islands. And looking at some of those results in a little more detail, I'll start with our flat island with a diurnal cycle, starting in the middle of the island looking at rainfall rate. Here on the y-axis, we have rainfall rate in millimeters per hour. On the x-axis, we have time in hours. This is averaged over all 5 days of our simulation. So this is essentially the average day of rainfall rate on the island. So looking a little closer, we have our cooler sea surface temperatures raining about an hour earlier than our warmer sea surface temperatures. And here in the secondary maximum, Our warmer sea surface temperatures last a little later into the evening and nighttime with higher rainfall rates. Looking over the coast, we have rainfall in the mid-afternoon, but the same is true that our cooler sea surface temperatures rain a little bit earlier and our warmer sea surface temperatures last slightly later into the evening. Looking at the ocean, it's clear that as sea surface temperature warms, we have higher rainfall rates. But I want to point out when I say higher rainfall rates, I'm talking about relative to this area, because here over the ocean, our highest rainfall rate is about 25 millimeters per hour, whereas in the middle of our island, we have about 1,000 millimeters per hour. So although these plots look similar, we're definitely losing a lot of our rainfall moving from the middle of the island outwards to the coast and to the ocean. I would also like to point out that the time of day rainfall occurs over the ocean is a little different than what we would have expected. Typically in a diurnal cycle, the rainfall over the ocean occurs in the late nighttime or early morning when the island is at its coolest. And so in a minute, I can explain why we think this occurred. Taking a look at cloud formations here, starting in the middle of the island. On our y-axis, we have height in kilometers. On our x-axis, we have cloud water and cloud ice concentrations in grams per kilogram. So we have low and high clouds here in the middle of the island. And actually, as sea surface temperature warms, we have less low clouds, but more high clouds. And so we think this is just due to their potential of growing much larger and much more convective when you have a warmer sea surface temperature. Then looking over the middle of the, over the coast and over the ocean, we still have our high clouds, less concentrations though. If you take a look at the x-axis, we're losing some of those high clouds moving from the middle of the island outwards to the ocean, but we're also losing our low clouds. But it's clear that as sea surface temperature warms, we do have increasing clouds, especially increasing high clouds. So we think we're losing these low clouds as you move out from the middle of the island to the ocean, because we have these large convective clouds here in the middle of the island. And as the wind pushes them outward, it forms a detrainment or an anvil. And as the air falls again over the ocean, it's sort of limiting our cloud formation here over the ocean. So a second ago, when I mentioned our rainfall rate occurring at a different time of day over the ocean than we thought, we think this detrainment is releasing latent heat, sort of stabilizing the atmosphere throughout the late nighttime and potentially the early morning. So we think the ocean doesn't really get the chance to rain until the late morning, which is why it occurs later in the day than we expected. We can look at the vertical velocities to support this. So here on the y-axis, we have height in kilometers. And then here on the x-axis, we have vertical velocity in meters per second centered around 0. So you can see in the middle of the island, we have a positive vertical velocity supporting the ascent of air in the middle of the island. And then over the ocean, we have a negative vertical velocity supporting the subsidence or the descent of air, supporting what we know about diurnal cycles in the tropics and maritime continent. And then taking a look at the mountainous island with a diurnal cycle, I'll start in the middle of the island again with our same rainfall rate plots. We can see that as sea surface temperature changes, we actually don't have a change in the initial time of day rainfall occurs, but we do have our warmer sea surface temperatures lasting slightly later into the evening. Looking at the coast, we have some varying rainfall rates at different times of day, and we think this is true because there's some complicated things happening between the rising of air in the middle of the island and the falling of air over the ocean. And then taking a look at the ocean, we have almost negligible rainfall, but it's clear that our warmest sea surface temperature has the highest rainfall rates. And again, I'd like to point out, when I say the highest rainfall rates, I'm talking about relative to this area, because we only have about 9 millimeters per hour here over the ocean, whereas we have about 320 millimeters per hour here in the middle of the island. Taking a look at cloud formations, we have the same plots as the flat island, and we have our low clouds and our high clouds still as well. And as sea surface temperature rises, we have more clouds in the lower and higher altitudes. Looking at the coast and the ocean, we begin to lose some of our cloud concentrations, but we do still have those high clouds, and it's clear that as sea surface temperature warms, we have more high clouds. So we think the same is true happening over this mountainous island, where we have large convective clouds in the middle of the island, and we have a detrainment or an anvil forming. And then as the air falls over the ocean, it's really limiting our low cloud formation and delaying our rainfall over the ocean into the late morning. And so we can look at the vertical velocities again to support this. Here on the y-axis, we have height in kilometers. And on the x-axis, we still have our vertical velocity centered around 0. We have a strong vertical velocity here at the surface, but we don't have much of a vertical velocity as you move up higher into the atmosphere. And so we think this is true because we just don't have as strong of a sea breeze convergence here on this mountainous island. So we don't have as strong of an ascent in the middle of the island. But then looking over the ocean, we do still have our negative vertical velocity supporting the subsidence of air. So looking at some concluding points about all of these results, warming the sea surface temperature can then increase cloudiness, which will increase the risk of heavy rain in these areas. This could possibly be due to increased water vapor and some plots that I didn't show in this presentation. The most rain fell in the middle of our islands, and in particular, the middle of our flat island, because we have the strongest sea breeze convergence there. Our warmer sea surface temperatures caused a delay in the secondary peak of rainfall, which can then cause it to last later into the evening and into the nighttime. Our islands favored large-scale ascent in the middle of the island, and our oceans favored large-scale subsidence, at least in close vicinity to our island where we took our readings. In the middle of our islands, we have high and low clouds, but as you move over to the coast into the ocean, we begin to lose those low clouds. So keeping all of these in mind, these become very important when you think about the daily weather and climate of the tropics and of the maritime continent and future decisions that might need to be made in these areas. Lastly, I would just like to take a minute to thank my mentor, Dr. Giuseppe Torri, for guiding me through this thesis project. I would like to thank the Global Environmental Science Program, including Department Chair, Dr. Michael Guidry, for supporting me in the completion of my project. And I'd like to thank my fellow SOAS classmates for collaborating with me on this work. I will leave you guys with those concluding thoughts, and I will open it up to questions if you guys have any. Thank you so much.
Excellent job, Kayla. Feel free to unmute and congratulate Kayla on a fantastic talk.
Congrats.
Great job, Kayla.
Congratulations, Kayla.
Solid.
Thank
Yes.
you.
Good job, Kayla.
Holy smokes, great job, Kayla.
Thank you. I think that's my dad. I'm sorry.
That's great. Let him go. He deserves this moment. Yeah. All right. Quiet time for questions for Kayla. Chris.
Yeah, again, it's more of a comment. I noticed in your mountainous island, of course, it's a simple structure that you build, obviously, because you have to do that. But I mean, what's driving the vertical velocities is the surface area that's heated in the island.
Yes.
So
yes.
Do you know what the ratio of your model island was to the flat island in terms of surface area, and how that would compare, say, to Oahu, where I suspect the surface area is much higher than your modeled area? And I just wonder if you have any comments about that.
Yeah, so we actually did the math, and the mountainous island has about 2% more of a surface area than the flat island. So not as much as we expected, but it added a little bit. And so we think this affected, I mentioned we had a little more, potentially a little more water vapor in the atmosphere above our mountainous island. And we think this could be why that is, because we have a slightly larger surface area that's heating, evaporating more water into the atmosphere. But compared to Oahu, since our island was so simple, just our simple bell-shaped curve, I do think Oahu would have a much larger surface area with a few mountain ranges. And so we compared our island more to like Sumatra or Borneo and equatorial regions that are larger, sort of with just one mountain range in the middle.
Yeah, interesting. Thank you.
Kayla, I have a question about the mountains versus the flat island. When Polynesians discovered Easter Island, they came upon an island that was different in many ways from their prior experience. And one of the differences that's cited is that it's flat. It's not a, quote, high island. So based on your model, I mean, you talked about the difference in temperature, island effect, but there's a mountain effect too. So how much can you give a relative sense of importance of the mountain versus the temperature effect?
Sorry, to clarify, your question is how big of a difference adding or taking away a mountain range would be to warming the sea surface temperature?
Right, exactly.
So I would say since we modeled 300 to 303 degrees Kelvin on both islands, we saw the most rainfall in the largest cloud formations in the middle of our flat island, because with our sea breeze convergence, when you have a mountain in the middle, it sort of slows down the convergence. And so that's why we didn't have as strong as an upwelling in the middle of the island. And so in our model, at least, our flatter island got the most rainfall and more clouds than our mountainous island at the same sea surface temperatures.
Really?
Mm-hmm.
Wow. Okay. Okay, I have another question for you. First, trivia. Hawaii gets more rainfall per annum than any other state in the United States. And the state in second place is where I'm sitting right now, Louisiana. Now, Louisiana happens to be smack on 30 degrees north latitude, which is where the Sahara Desert and the deserts of the southwestern US are found. Why do you think Louisiana gets so much rainfall?
Unrelated to this study, I would say it might have something to do with being so close to the Gulf Coast and having so much water next to it available for evaporation and cloud and rain formations. But I don't know if I can support that.
No, I think that's probably the answer. Yeah.
I was going to say the Cajun effect, but.
Kayla, I had a question for you.
Yes.
You mentioned earlier in your presentation there was a third island you didn't have time to discuss. I was wondering briefly if you could just tell me what the third island was and what you found out about it.
Yeah, so that was actually our first island that we created. And when we were setting up the domain of the island and starting the first simulation, we hadn't imposed a diurnal cycle on it quite yet. And after running the simulation, we found that we had a horizontal jet about 5 kilometers up, pretty strong. And so we thought this might skew some of our rainfall rates and cloud formations that we'd find when we imposed a diurnal cycle. So we changed the domain a little bit, shrunk the island and lengthened it a little bit. And then we lost that horizontal jet in these runs that we did. So we kept that island in our study so that we can compare a diurnal cycle island with an island that sort of has some random heating and cooling with no diurnal cycle. But we didn't think it was important enough to include in this presentation.
Awesome, thank you for speaking on that.
Yeah.
I had a question.
Yeah.
I was wondering where, if there is any place that we actually see sea surface temperature changes like this currently, like in the real world.
Yeah, so we actually can see sea surface temperature changes like this in El Nino or La Nina years in the maritime continent. And so we can see these actually happening, increased rainfall rates and cloud formations. And so this, like I said, this more simulated equatorial regions in the doldrums, we had no background wind imposed in these models. So areas in the ITCZ where there's no trade winds, which is why simulating Hawaii would have been very different because we have our trade winds. And so we would have seen some different effects if we imposed some background wind in the model.
Time for one more question. Okay. Well, excellent job, Kayla. Well done.
Thank you.
This concludes the Spring 2021 GES Symposium. We thank everybody for joining us. Thank the presenters for doing a fantastic job, and also the mentors. Without them, none of this would have been possible. So thank you to all parties involved. So we will hopefully see you in our summer presentations, which will be in August. So we'll make sure that everybody's aware of those and gets the information on how to connect. Otherwise, again, thank you for joining us. If everyone could unmute and congratulate the speakers, that would be great. And that concludes our symposium.
Great job, everybody.
Congrats, everybody.